Hi guys, we are working on section 2, passive transport across a membrane. So first we're going to start discussing what a solution is. It is made up of two parts, the solvent and the solute. The solvent is the liquid, it is the thing that does the dissolving, and the solute is the thing that is going to be dissolved. So in salt water, water is the solvent and salt is the solute. Diffusion is the net movement of molecules down a concentration gradient. So moving from an area of high concentration to an area of low concentration. Think about when you spray air freshener. If you spray it in one spot, it takes a long time for it to move throughout your room. It moves from an area of high concentration on one side to an area of lower concentration on the other side. Molecules move both ways along the gradient, but the net movement is from high to low. Equilibrium occurs when the net movement stops, so they're moving at the same rate from one side of the room to the other. And it is when the solute concentration is uniform, so there's no gradient. So it's like after I have finished stirring the sugar cube into my coffee. When I first put the sugar cube in, it is going to diffuse from an area of high concentration in the bottom of the cup where the sugar cube is to areas of lower concentration like the top of the cup where there's not a lot of sugar. When the concentration is uniform, the cup of coffee will be sweet all the way throughout. If I put too much sugar, the coffee can only dissolve so much sugar and it will settle to the bottom and it will not reach equilibrium. Temperature, pressure, electrical currents, and the molecular size affect the rate of diffusion. Small molecules diffuse much more quickly than large ones. Temperature, hot things spread out faster. So if I were to dissolve sugar in hot water, it's going to dissolve faster than in cold water. That's why when you make sweet tea, it's important to put the sugar in while it is still hot, so that it is easier for that sugar to dissolve, to become uniform throughout that pitcher. So here we have a cube of crystal dye in a fish tank, and it starts out at a very high concentration right here in the cube, and a very low concentration of dye up here where the fish are. Over time, the dye starts to spread out. Okay, they are going from an area of high concentration to an area of low concentration. Over time, we will get an equal distribution of water and dye throughout that fish tank. Now, I don't recommend putting dye in your fish tank. It might kill your fish. So, best to leave this just in the diagram, guys. Gas exchange in your lungs. So, this is a lung. Okay, this is a bronchiole, and down here we have an even smaller view of alveoli, and they are used in gas exchange. So basically, the oxygen that you breathe in is following its concentration gradient from an area of high concentration in the air that you have just breathed in into an area of lower concentration in your bloodstream. And it's the exact opposite for carbon dioxide. There's a high concentration of carbon dioxide in your blood, and it wants to go to a lower concentration in the air. So the oxygen and the carbon dioxide just switch places. Another type of passive transport is osmosis. This is a special case of diffusion that focuses on the movement of water. It is the diffusion of water across a selectively permeable membrane. That is the standard textbook. Everybody knows that definition of osmosis. So when the solute concentration on one side of a membrane is high but the water concentration is low, the water is going to move. If the solute concentration is low but the water concentration is high, the water is going to move. The water can diffuse across that membrane, but the solute cannot. So the net movement of water is toward lower water or higher solute concentration. So it's going to move to an area where the solute concentration is higher. The way that I remember it is salt sucks. Salt draws water toward it. And that's why if you drink salt water, you get thirsty, because salt sucks. It's going to draw water toward it. Osmotic pressure is the pressure that develops due to osmosis. That kind of makes sense, right, guys? Here is a demonstration. Okay, here, this red line is a selectively permeable membrane, meaning that water can move through it. So we have less water in our container. Okay, so we have a high solute concentration. And so water is going to move into our vessel, into our container, trying to reach equilibrium. All right, we're going to do a lab that's kind of similar to this. So just hold tight, and we'll probably get to it early next week. At least that is the goal. So ionic solutions, that is when the solute and the water concentrations are equal. Sorry, I said ionic. Isotonic, there we go, that's the word. Isotonic, solute and water concentrations are equal on both sides. So whenever there's movement of those molecules, there's no net movement. The same number are moving in and out at the same time. A hypotonic solution is when the concentration of solute in the solution is lower than the inside of the cell. When the cell is placed in a hypotonic solution, it will swell up. And this is what causes turgor pressure in plants. It's what causes them to be very crisp and crunchy, and gives them that nice, like lettuce, that almost watery taste. Plant cells, they don't burst as easily as animal cells because they have that cell wall that stops them from absorbing any more water. Animal cells, they don't have that, so if they take in too much water, they will lyse or rupture, and they'll just burst. Protozoans living in freshwater environments, they have something called a contractile vacuole that is able to rid them of excess water. So whenever they absorb too much, it's kind of like whenever you squirt water out of your mouth, like whenever you're at the pool and you like spit water at somebody. That's kind of what it looks like. It's kind of cool to watch. I'll see if I can find a video and show it in class tomorrow. Hypertonic solutions are when the solute concentration in the solution is higher than inside the cell. So if you place a cell in a hypertonic solution, it will shrivel up and shrink. It's called crenation in animal cells and plasmolysis in plant cells. So here we have some diagrams. Isotonic solution, that's when cells are happy. Things are moving in and out at the same rate. There is no net movement. In a hypotonic solution, water enters the cell and it can burst. You'll notice that the central vacuole fills up and takes up a lot of space in the plant cell. That's because they don't know when they're going to get water again. So they're going to absorb as much of it as they can and hold on to it. If you were to place a cell in a hypertonic solution, water is going to leave the cell. It's going to exit and your animal cell is going to shrivel up, called crenation. And in a plant cell, the plasma membrane will actually pull away from the cell wall and it'll shrivel up inside, and this is called plasmolysis. And it's not good for the cell. This is what happens when you forget to water your houseplants, or you forget to put water in that vase of flowers like I did, that I had sitting on my end table for 3 weeks, and they all just kind of shriveled up and died and looked really gross. I wish I was lying, guys. It sat there for 3 weeks. Another type of transport, facilitated transport. This is when the molecules need some help getting through that membrane. So these molecules must combine with specific carrier proteins to move across the membrane. They can't just join with any of them. They need to find a specific protein that will allow them to move, and they follow the concentration gradient, moving from high concentration to low concentration. So it's not active, it doesn't require energy, but it does need the help of a protein. And that protein is there to make sure that it does not get stuck in that phospholipid bilayer. So here we have a carrier protein. Here is our solute, the thing that is wanting to go from high concentration to low. It will bind with that carrier protein and move through. It doesn't require energy, but these molecules are too large to pass through the membrane without some help. And it will happen over and over again until we get equilibrium. They can move either way through the membrane. So if we had more of them inside the cell and they needed to get out, they could do that too. All right, so that is it for this section, guys. If you have any questions, let me know.